Without wasting more time, because we have so much to do, I'm really excited about the next panel for so many reasons, right? Neurodegenerative disorders and Alzheimer's disease is such a pressing problem. There's been so much amazing preclinical research and new emerging clinical trials in this field with focused ultrasound. So very, very excited. I would like to invite all the panelists who are coming up already. Great. And we are really, really fortunate today to have as our moderators George Vredenberg and John Dwyer. George is the co-founder of Us Against Alzheimer's, and John is the president of Global Alzheimer's Platform. They are a dynamic duo who work together often. And I think what's great about having them here as moderators today is we do all this really innovative and groundbreaking preclinical research and even clinical trials, but they see this problem in a much broader concept. So they're going to really help us figure out how we can take our knowledge and really advance it to patients and help people. So I'd like to formally introduce George, who's probably going to speak first, and he'll take over from here. So George and John, thank you for coming. And to the rest of the panelists, let's have a good time.
I heard this was the best party in town, so we'll have a good time today. My name is George Vredenberg. I'm the chairman and co-founder of the more aggressive of the two patient advocacy organizations. My organization is called Us Against Alzheimer's. So we run to the toughest problem that exists in the Alzheimer's space at any point in time, and so we're running a fair amount these days, but today is a sort of a changing time in the Alzheimer's space because we do have one approved and soon to be a second approved drug in the Alzheimer's space. My partner, John Dwyer, who's the president, he's another co-founder of Us Against Alzheimer's, he's also president of the Global Alzheimer's Platform Foundation, which is the only global platform for clinical trials in the Alzheimer's, Parkinson's, and CNS space. So John will speak in a minute. He and I are co-moderating. We're really excited today to have a real esteemed panel, many of whom, probably all of whom you all know well, but Jürgen Getz of the University of Queensland, Alexander Carpentier, I thought she was going to be up on screen, but she will be. Jinwoo Chang of the Yonsei University College of Medicine, Ali Rezai, did I pronounce that right, Rezai? Of the West Virginia University Rockefeller Neuroscience Institute, and Sandra Black from Sunnybrook, all of whom are well known in this field, experts in this field, and so you'll be entertained by them. So I will say just a few words about where we are in this space. John will speak a bit about how focused ultrasound might be relevant to what's going on today in the Alzheimer's space. Then we'll have brief comments from each of our panelists, and then we'll have hopefully a lot of time for questions. As I said, this is an interesting time, a real changing time in the Alzheimer's space. The Biogen drug aducanumab was approved in June of 2021. And Medicare decided not to cover any of the monoclonal antibodies. So the marketing of aducanumab was pulled as a result. We've recently had very good results from Eisai's drug, lecanemab, which demonstrated an ability to reduce amyloid and tau in the brain, and as a consequence, demonstrates some clinical benefit, a 27% reduction in the rate of decline, which for patients means a couple of years worth of functional stability at an MCI or mild Alzheimer's stage. So for the patient community, this is exciting. A lot more evidence will come out, and details and data will come out at the CTAD conference at the end of November in San Francisco, and so we're all looking forward to that. The trial was large, 1,800 people. It was highly diverse in terms of racial composition, gender composition, and comorbidity composition. So we'll be able, hopefully, to be able to see who were the better responders and who were not responding as well. So a 27% rate of improvement in the rate of decline means that some people may be performing significantly and responding significantly better than that, and some people may not. So hopefully the greater data on this drug will demonstrate in whom and which subpopulations the drug is most effective. Safety seems to be manageable, significantly less ARIA-E and H, as in the prior drug, still some ARIA concern. The challenge with this drug, as with all monoclonal antibodies, is that they're infusion products, at least at the outset. Twice a month for the ASI product. For the follow-on drug from gantenerumab from Roche, whose results in the phase 3 trial will be announced in a couple of weeks, we'll see whether or not that drug proves to have the same kind of or better than the level of clinical benefit shown on lecanemab, and donanemab is coming next summer. Now, the challenge is obviously we have an infusion product twice a month for the ASI drug. They are moving on to a subsequent follow-on trial to demonstrate whether or not they can do once a month. They're following on with a subcutaneous version to try and reduce the burden on the patient in terms of administration and ease the access to it. With that said, these drugs are still very expensive. And indeed, there are going to be a slow rollout, even if Medicare changes its posture and decides to cover these drugs. So that's where focused ultrasound may come into play, and John will talk a bit about that. Now, I put this particular slide up because the path to actually getting a device or a drug onto market has a 10 to 15 year, at least a 15-year sort of cycle to it. It's a long path. And so thinking about focused ultrasound as both a research project, preclinical, but getting it into clinical trials, demonstrate safety and efficacy, and then beginning to work through all of the stages of trials in humans is a non-trivial task, as you all know. But then you have FDA approval, then you have Medicare, and then you've got to train a healthcare profession that is really not yet attuned to interventions with respect to Alzheimer's. So every one of these steps is important because we on the patient community, we care very much about your doing the research. We care very much about getting your research into humans and creating a clinical benefit for all of us. The other point I'll make, in just the next slide, is the fact that we have a diverse population that is going to take these drugs. Do you have the next slide, please? Or not. Which means that, in fact, the racial minorities who have a greater propensity for these diseases we have to make sure that we get any intervention in the face of Alzheimer's into all the populations. This happens to be a slide that shows how on the left-hand side, where the genetic research has been done in Alzheimer's, 90% in white Caucasians. The right-hand pillar is the distribution of populations around the world with Alzheimer's. And so this is a global picture, but in fact, in the United States, a significant percentage of the population experiencing this disease disproportionately is Black and Hispanic. So as we think about getting these interventions to patients, we have to think about inclusion, equity, and thinking holistically, as well as getting the price down, getting the accessibility of the intervention made easier, and as a consequence, we're going to have a good panel discussion about the research, but about getting the product actually into the minds of those that are deeply affected by this disease. So with that, I just want to introduce my co-moderator, John Dwyer, who will speak a bit about focused ultrasound and its applicability to these monoclonal antibodies. Thank you.
I come to these things and I say, why am I here? Looking around at the level of MD, PhDs, and general clinical knowledge assembled here, and I last took a biology class commensurate with Darwin getting off the boat. So as a consequence, I'd like to have my moderating remarks focus on what George said in an area that I have some expertise, which is building businesses and accumulating capital to go after new markets, which is clearly what focused ultrasound is, from my way of thinking. In the area of Alzheimer's disease, there have been in our organization, a word about our organization. So the Global Alzheimer's Platform, as George alluded to, has a very simple but somewhat ambitious goal to reduce the time, reduce the cost, and substantially increase the efficacy of Alzheimer's clinical trials for everyone. We've been in 15 or are concluding 15 phase 1, phase 2, phase 3 Alzheimer's trials. We're conducting those with the major sponsors. So my remarks are anonymized by any one specific trial, but my observations have an N of 15 for those of you that ask those kind of questions. Roughly speaking, when the CEO of Medtronic comes up here, tell him that on the average, an Alzheimer's clinical trial will spend roughly on a phase 2 or phase 3, over $100 million can be clearly articulated as the time spent to titrate the drug in the form of a monoclonal antibody so that it gets uptake into the brain and minimizes adverse events. Your technology, if I believe what I read, and I believe what I read, would be an enormous benefit to rebalance clinical benefit with reduced dosing of the drug and similarly providing lower adverse indications associated with the drug, which George aptly described lecanemab. Every monoclonal antibody in Alzheimer's is going to have a conversation with the regulators about benefit versus the incidence of ARIA in that population. It is my thesis that your technology, what you folks are focusing on, no pun intended, is a remarkably promising solution to changing that balance. The amount of investment, the good news about lecanemab is it has great data. It will circumvent much of the discussion that Biogen's Aduhelm generated, and I can tell you with a high degree of confidence that the Alzheimer's clinical world is in a good place for the first time in many, many, many years. So the opportunity to bring your technology forward and have it become a useful, direct, clinical benefit enhancing, cost reducing technology for all the sponsors and biotechs that are bringing technologies forward is very real. I might add the untested frontier here is the use of your technology to change the whole discussion around development of tau targeting therapies, which have not gone through anything close to what beta amyloid targeting therapies have in terms of multitudinal trials, dosing studies, and they need to compress their timeline and improve their benefit to adverse indications ratio as well. I want to say one more thing about your technology I find appealing, and that is that we spend a lot of time in my organization trying to figure out how to conduct clinical trials where the poor, the underserved, the higher disproportionate incidence of Alzheimer's disease exists. And the opportunity to bring drugs and deliver them with a combination of a digital cognitive test, a blood-based biomarker, focused ultrasound, deliver the drug without a PET image, deliver the drug with a lower concern about adverse indications is extremely attractive when you're worried about being in the Rio Grande Valley where there is no PET machine available for 320 miles capable of doing a beta amyloid or tau PET, and the likelihood of your equipment being able to be brought together with an MRI and deliver a solution that isn't going to be similarly hampered is really attractive if we're really going to bring Alzheimer's therapies that are disease modifying to the folks that need them most. The county that we're in Texas, is always annually or every census nearly the poorest, highest incidence and prevalence of Alzheimer's in the country. And we can't get today's DMT technologies in any reliable way for 5, maybe 7 years from now. So it's with those remarks that I'm very happy to be here, eager to learn more about your industry, eager to help support you all in your endeavors to bring your technology to a disease state and field that needs it, and we look forward to going forward with the program from here. Thank you.
Thank you, John. Let's start off with Jürgen Getz to lead us off with some real expertise here.
I think I got 2 slides. Yeah. So I'm an Alzheimer researcher, and I think that's the second slide. I got one. Let's see. How does that work? Yeah. I'm an Alzheimer researcher, and I entered the ultrasound space only recently. And so we have, over the years, we have been making transgenic mouse models, and we treated these mice either with ultrasound on its own, or we also used ultrasound with microbubbles, and we also developed our own anti-tau antibodies, and we have also been trialing aducanumab. And we always found that the combination trial worked better than the individual treatments. What I would like to capture with this slide is that, in a sense, so we have been treating between 4 and 14 times weekly. But that holds true for us, but also for, I guess, most groups working in the space, is that we only look at immediate effects. So we basically, after we had the last treatment, we sacrifice the mice and then we analyze them. But what's really not known is what the long-term benefits are. And I think this is something which is still lacking. Another aspect which is highlighted in this slide is that we tend to focus on A-beta and tau, but ultimately we want to achieve cognitive improvements. And we have seen this with the aducanumab antibody, that there was, in one of the 2 trials, that there was removal of amyloid, but this was not sufficient to lead to cognitive improvement. And in a sense, when we look at the field, it has been, and how it historically developed, it has always been guided by neuropathological findings. So focus has always been on A-beta and tau, but in my view, there's a dissociation between A-beta and tau clearance and cognitive improvements. There's another aspect I would like to make in my next slide, and that is, and I can hardly see it from here, is that when we want to treat Alzheimer's disease, we are dealing with a range of problems which are specific to Alzheimer's disease. One is a lack of robust biomarkers. Another is also that the brain is, in a sense, inaccessible, something we know can be overcome by ultrasound. But also another aspect is that there's no clarity about which brain area to treat. When you look at the right, you see in the slide that when we look at the A-beta and the tau pathology, for example, the tau pathology starts in the entorhinal cortex and spreads into cortical areas and spares the cerebellum, whereas the A-beta pathology again has a different pattern. So it all depends on the time point at which one treats. So we see on the one hand that A-beta and tau have different patterns, and these patterns don't overlap, and on the other hand that there is a spreading. So one question really is when to treat. And when you look at the left, you see that A-beta and tau come as different species. Tau and A-beta are post-translationally modified, there are splice variants, and again, the question is which species to target, and it's still not clear at present which of the A-beta and tau species is really the toxic species. So basically we are dealing with a few fundamental problems which makes it really difficult. And the final statement I would like to make, just based on our own research, while on the one hand we know that dementia is more closely linked to tau pathology, on the other hand we have found that tau is a much harder target to clear.
Thank you, Dr. Getz. Alexander Carpentier, who's here virtually.
Well, I'm here to talk about the little trial, phase 1 trial we did using the SonoCloud device on Alzheimer patients. Well, I don't see correctly this slide I sent you, so I'm going to upload it on my computer. But it was a pilot safety study performed on Alzheimer's disease patients with 7 repeated blood-brain barrier opening sessions with no associated drug. The goal was to see safety. And as you know, I am working with contact ultrasound, so it's an unfocused, non-focused ultrasound in order to address the cortex maximum and a larger region. So the device was only 10 millimeter diameter, implanted under a local anesthesia procedure in an out clinic fashion. And we had actually 63 sessions for all the 9 patients that went perfectly correctly with no adverse, even no toxicity. So we were expecting to have actually a significant decrease of the amyloid plaque under the system in the acoustic field. We found that it was somehow the case, but it was not statistically significant that much. So we were a little disappointed. And the goal was to increase the number of sonications in order to be sure that a repetitive treatment would have a better effect than a one-shot or whatsoever treatment. And we found definitely an effect, but as you see on the different curves, on the left side is the amyloid PET analysis that shows a decrease and afterwards somehow plateau. And the other one was FDG, because we didn't have at that time the collaboration with the pharma who does the anti-tau PET analysis. So this study is interesting, not necessarily because of the results, but it's interesting in terms of efficacy, at least on PET studies. But it is interesting in terms of safety in patients with Alzheimer's disease by doing repetitive sonication in an eloquent area, because it was based on the gyrus supramarginalis on the left side. So this is my slight contribution. My main concern in the future, I'm actually fighting a little to find some pharmaceutical company that would want to do a phase 1/2 study with, for example, an anti-tau, and hoping there to boost the drug itself, but also boost the effect of the ultrasound. Voila.
Thank you very much, Dr. Carpentier. Jinwoo Chang of Yonsei, please describe your research, your work.
Can I have my slide? Go back. Okay, I underwent the phase 1 BB opening trial for glioblastoma 4 years ago. At the time, I've used the primitive, a little bit, the earlier type of the software of the InsightTech. But for the second clinical trial for recurrent glioblastoma, we used new software from InsightTech. We could open a more extensive area compared to the first one. So I applied the technique for the Alzheimer's disease, and we underwent the phase 1 clinical trial for Alzheimer's disease. We opened twice with 3-month interval, and we also underwent the FDG and amyloid PET for this patient. Interestingly, we demonstrate reduction of the amyloid plaque and the improvement of the emotion, because we opened the BBB in the frontal lobe to confirm the safety issues. main difference with my clinical trial compared to other group is we open more extensive area. Other group just open 1 or 2 or 3 cubic centimeter, just 1 or 3 cc, but I opened almost the 30 cc's in total in the frontal lobe. I didn't see any complications, and based on my first clinical trial, recently I'm now undergoing the second clinical trial with same protocol. Main difference is we open the BB opening 1 additional time, 3 times with 2-month interval. At this moment, no one knows. The previous speakers just opened the 7 times with 4 months interval. In the animal, weekly or bi-weekly be opening for the session of the 15 or sometimes less than 10. But for human, no one knows. But I just want to open the extensive areas and want to confirm the safety issues. At this moment, I didn't want to add the drugs because the pathology of the Alzheimer disease is quite different and quite universal, global disease. So I just confirmed the safety issues. If most interesting things in the middle of treatment is we start to use the transducers, and nowadays we don't shave the hair of the patients. So there's no complaint and no problem with the patient compliance. So I think if we can find the benefit of the BB opening alone, maybe this technique can be useful in the clinical field in the future. Thank you.
Thank you very much. It's fascinating how promising this all is. So we're still in somewhat the early stages, I would say, but nevertheless all promising. Dr. Rezai, would you like to offer?
Thank you. From our perspective, Alzheimer's, as Dr. Chang was saying, that it's a neurodegenerative condition, different pathology than a brain tumor, than a thalamic lesion for tremor, HIFU, or other lesions. So first, it's important to determine safety in a chronic progressive condition. Second, I think ultrasound is important because it can target anywhere in the brain, superficial, deep, around vessels and others. With safety, no head shave. Now we're using no frame. 80-year-olds are coming in to the outpatient MRI, going home the same day. I think that opens up the opportunities. So I was going to talk to you about the trial that we've been doing. This is an Incytec-sponsored trial, 4 years now. 18 patients enrolled at Rockefeller Neuroscience Institute, Ohio State University of Vibor Krishna, and Mike Kaplan at Cornell Weill Medical College. FDA, IDE, the goal was safety. We started with 1 cc's in hippocampus first, and over time, FDA gave us permission. Now we're doing 40 cc's. So we've gone to 40 times more volume. Entorhinal cortex, hippocampus, then we went to the frontal lobe, parietal lobes, superficial, deep, all different areas. And we're looking at, you have to reduce the biomarker, reduction beta amyloid. You have to demonstrate that in the targeted region, and also evaluate cognitive and neurological function. Trial design, baseline, imaging, clinical, 3 different treatments, 2 weeks apart, each of them, and then follow up at day 7 after the last treatment with beta amyloid PET, and it's a continuous trial. Quickly, results, you can see the hippocampus on the left side, panel A, that's the target for the FUS. And then B is contrast scan right before you sonicate. You deliver microbubbles and sonicate. C is immediate BBB opening, demonstrated by gadolinium going into the interstitial space from the blood, because the BBB is open. And then D, 24 hours later, the BBB is closed. Same thing, you can look at panel E, frontal lobes, superficial and deep part. The arrow shows the areas that are sonicated, immediate BBB opening. Contrast goes into the brain interstitium, parietal areas and outside, and it resolves within 24 to 48 hours. So in this initial effort, FUS is safe and opens the BBB in a conformal fashion, only where you treat, not outside. Around cortex, subcortical, ventricles, blood vessels, it seems to be safe. We've noted no overt hemorrhages, neurological AEs, or SAEs related to the procedure. The opening was on demand, transient, and reversed in each case within 48 hours. In the long-term study that's in press right now, Journal of Neurosurgery, there was no deterioration in neurological or cognitive function versus the ADNI comparator group at 1 year plus. and there was a focal reduction in the targeted areas of 5% SUVR and 14% centilloids with just FUS-BBB opening. And now this is an opportunity for either deliver FUS, as Dr. Chang was saying, open the BBB, you get clearance in amyloid, or combine it with Aduhelm, donanemab, lecanemab, and that's the future. So we initiated the clinical trial now, combining Aduhelm with FUS-BBB opening. We've enrolled one patient, and we're enrolling the second subject in that trial. Thank you.
Boy, you panelists are really efficient with your time. It was really very impressive. Dr. Black, would you like to report on your understanding of the applicability of FUS to Alzheimer's disease?
Yes, so I had the pleasure of being on I'm an investigator in Alzheimer's disease trials the last 30 years, very actively involved in the current trials with the disease, hopefully disease-modifying therapies, and excited with some of the progress that's happening in our field. But I had the pleasure and privilege of being part of the team that did the really first-in-man focused ultrasound to see if we could safely open up the blood-brain barrier. We were very cautious and decided to choose the right frontal lobe, which if you injure, it may not affect the person's life so dramatically. And so if we can see the next slide, perhaps. This is from the Hartwell Neuromodulation Center, Harvard's Brain Sciences Research Program at Sunnybrook Health Sciences Center, and the director is Dr. Neil Lipsby, who's the neurosurgeon who's really driven all this, and Dr. Meng has been his right-hand trainee, and I'm there as the expert. If we could just do the first slide showing the focused ultrasound setup. Yeah, so this was our setup, and in fact, people had to be in a frame. We did 5 subjects, we followed them first of all weekly and then in a month, and then we actually added on another one. It's all published in Nature Communications in 2018. And what this shows is that we were opening up the blood-brain barrier. This shows you the contrast, the gathering contrast after the procedure, and it was gone the next day. But clearly, we're actually seeing perivascular spaces, probably removing the contrast, and hopefully we'll be removing the amyloid, because we know the amyloid goes along the perivascular spaces, microglia processing, and also just in deep sleep, you are getting rid of the garbage of the previous day, including amyloid. So that's very important. Now, the next slide shows, one case where it wasn't everybody, but it did look. If you look at this PET scan, we can see the uptake in like the sort of yellow area at the front, and there was some, decrease in it over time, which was kind of a nice surprise, but wasn't replicated in everyone. So I think there's going to be variability in who's responding and what stage they're at. This person did have some asymmetry in his pattern of amyloid uptake, but we were pleased to see in this person that actually we were able to remove, at least reduce what looked like positive amyloid. In a later study, we also were interested, if you see the next slide, we were interested in whether or not there might be some other effects, which I think is going to be very, very interesting, because it's not just that you could maybe couple it with an antibody, or we've been interested in IVIG, which is a natural product, or you have auto antibodies that you can get in more efficiently with opening up the blood-brain barrier. But we also looked at resting state fMRI, and there was a hint that we were actually slightly modulating the functional connectivity, which opens up the possibility that when you are doing this opening, you might also be affecting circuitry that hopefully could be manipulated in a desirable fashion. So our next step was to do multiple openings, and we have started on that journey with the CAMPL and default mode in particular being areas of targeted opening, using focused, the ultrasound to be able to do so. And unfortunately, it got interrupted very seriously by the COVID pandemic. And so we had to put things on hold, but we got a few people in, and we're going to be continuing to do that, potentially also including with it some of the IVIG. We don't have access to aducanumab. It has not been approved in Canada. But there will be other antibodies. I think that lecanemab is quite promising. And so that might become more possible for us to couple the antibody, with ultrasound. And my understanding, and the basic scientist can correct me, I think it's something like 0.1% or 0.5% of the antibody that actually gets through the blood-brain barrier without assistance. So the other idea would be, if you could do this less and less invasively, and we're interested in developing a helmet that could be personalized for each person with their key areas being targetable. It might be that you could more efficiently use these antibodies, which are quite expensive at the moment. And so that would be a more efficient way to take advantage of what developments there are in antibodies that might work as disease-modifying therapies. So those are some directions in which we're going right now. It's to gear up and continue the multiple openings, but we've had to restrict that quite a bit, at least at our site in Canada, because of the COVID pandemic.
Thank you, Dr. Black. As I say, all of you have been extraordinarily efficient. Let me just ask a question, then John will ask a question, and then we'll go to the audience. As we've seen, the trials that simply reduced amyloid generally were not very successful. But it turns out that targeting now on some of the second generation of monoclonal antibodies is very precise at a toxic form of amyloid. I'm curious to see, and that has proven to potentially to show some real clinical benefit. So my question is whether or not the focused ultrasound will enable us to target particular epitopes of amyloid and potentially tau, or is this a general reduction in amyloid you're seeing? And what evidence is there to date? I recognize we're still in the early stages of clinical benefit from the nature of the reduction of amyloid that we're seeing in those very early signals. So perhaps Dr. Getz, maybe if you might take that, and we'll come to the other panelists if they have a comment.
Yeah, thank you. I think, we have been targeting both tau and A-beta, but as I've said, I think tau is a much harder target for a few reasons. I think there's a discrepancy in the field as to whether tau is mainly extracellular or mainly intracellular. But then again, the extracellular tau is either available as free aggregates, which would then mean that it's accessible, or it may be enclosed in exosomes, which means it will not be accessible. Now, when we think of tau as an intraneuronal target, which is my viewpoint, then I think compared to A-beta, it's a much harder target because any therapeutic agent, an anti-tau antibody, not only needs to pass the blood-brain barrier, it also needs to pass the extracellular matrix, which has its own pore size of between 30 and 60 nanometers. It needs to be taken up by a neuron, it needs to engage with its target, and it needs to be effectively cleared. So in my view, tau is a much harder target as compared to A-beta.
And what is the evidence to date of clinical benefit? Maybe Dr. Black, you want to take that?
Well, if I could just comment on this, because I think, first of all, the concept we have to have is that there's a latent period of accumulation of these proteinopathies, these different proteins that deposit and become injurious. There's a concept that they're there maybe as a protective response to something, as you know, like infections, very relevant today in COVID today, or like head injury or stroke. You get a buildup of amyloid that sort of walls off the area of injury. And amyloid and tau are kind of a tag team. So amyloid is kind of walling it off, and tau it's actually injuring the synapses, killing synapses. Tau is cutting the axons, and then the microglia clean up the mess. So we're seeing probably a chronic immune response syndrome of some sort, and the amyloid and tau are a bit of a tag team. And I think it's still reasonable to try to do something with the amyloid because that seems to be more engageable with the antibodies that we have to date. The idea is that the tau then sort of spreads both extracellularly, which you described very worrisomely, I must say, but also intraneuronally. Once it gets going, it continues to seed. And eventually what happens with the amyloid is it builds up, but it actually plateaus, and then it starts to go down. And some people think that's because the brain is shrinking and there's not a cortex to contain it. So I still think amyloid is a reasonable target, but getting rid of it with these antibodies is, brings some risk, and depends also on the health of the vessels, and that's why vascular health is so important and physical health is so important for even how you would tolerate these drugs. And then we've got the characteristics like the ApoE4, genetic characteristics that make you at greater risk of having a leaky blood-brain barrier to start with. And then there's autosomal dominant disease where you've got a mutation that is building amyloid from birth. You're laying down amyloid from birth, so you start to get sick in your 20s, whereas older individuals will start to get sick in their 60s and 70s. So I think it's still reasonable, and we're moving, as you may know, toward what we call the pre-symptomatic stage, where people may be at risk. Now we're using the blood tests or CSF to know that the person probably amyloid depositing in the brain through a phospho-tau isoform. It could be 181, 217, or 231. That is being used by the trialists now to screen the person to make sure that they actually have the disease. Then they go on to the PET scan, the imaging, and the rest of it. And so I think we're getting to a more precision-oriented approach, and the next few years are going to tell us whether it really does work to slow people down, and whether it can even be used in the pre-symptomatic stage where there's less damage done to the networks, hopefully. And we'll see that soon.
Great. John, you got a question?
Yeah, a different point of view. If I was CEO of Medtronic, who might be here after lunch, the question I think that all of you should be in a place to think about is, what are the regulatory opportunities and challenges facing the effective delivery of this technology, particularly with the FDA and CMS?
Yeah, I think the key thing here is safety in terms of looking at Alzheimer's patients. But I think the key thing from my point of view is combining this with the pharma and the biologicals. And it's non-invasive. The MRI becomes a therapeutic tool, no head shave, so it opens up a new opportunity to target anywhere in the brain, depending on whatever the biomarkers, doesn't matter, beta amyloid, tau, and get people pre-Alzheimer's, MCI. When they're getting Alzheimer's, it's too late, in my opinion. And the other problem with the pharma is that the doses are very high, and about a third of the patients have ARIA-A's, ARIA-E, ARIA-H. So if we can reduce the need for high doses and deliver a lower dose of antibody into the area specifically that you open, so you can get a reduction in the biomarker, and within a few months versus 18 months, I think that's the opportunity. And we need the pharma to work with the ultrasound community to get this going faster. It's going way too slow. For our trial with the Aduhelm and ultrasound, it took us 1 year to get FDA approval. Back and forth many times, all for safety. So this needs to go faster.
I have a somewhat different opinion about clinical trial combined drug, especially recently even in the animal models. If we combine aducanumab, there was an improvement of the memory. But for human, if we want to combine with aducanumab with ultrasound, we need to cover the whole brain. It's impossible. At this moment, if we only combine the aducanumab for one certain area, it may be difficult to get the real benefit. So I think with better you combine other drugs, but that is my estimation. At this moment, we also need to confirm the real benefit of the ultrasound itself. If we confirm the real benefit, as we demonstrate in the animal experiments, we can combine aducanumab or other drugs later on. But at this moment, it's too early to conclude the result with the drugs at this moment.
I would also like to make a comment. Is this on? Yeah. And I think it might also depend on which brain areas one wants to treat. I think one needs to differentiate between brain areas which have amyloid and those which have not. Because you have, for example, shown that in senescent mice that ultrasound, even without microbubbles, activates the NMDA receptor. Now we know that in Alzheimer's disease, a beta overactivates the NMDA receptor. So kind of what's good in a physiological context may be damaging in a pathological context. So I do think that one needs potentially to rethink how one treats different brain areas depending on whether we have a pathology or not.
I think we ought to open up the floor to questions. If anyone in the hall has any questions, we'd be delighted to hear from them. And until we get a question from the audience or online questions, okay, none. Dr. Carpentier, do you have any reaction to the comments that have been made in terms of where the best potential use of focus ultrasound is going to end up?
Yes, actually, you said totally correctly that we have now some drug that goes without ultrasound that have sufficiently enough efficacy in reducing the amyloid. So the key question is tau, and will decrease of tau be useful in terms of cognitive impact or not? And I think that ultrasound may help to have a sufficiently enough concentration of anti-tau antibody inside the brain, and that will get into the neuron, actually. And considering low delivery of antibody in the brain, you have an additional low antibody in the neuron. So maybe ultrasound will help this gradient to be bigger from outside the neuron to inside the neuron. And this is, I think, maybe the place. The second, for a high view and ultrasound. One other key question also is to put in the brain, thanks to ultrasound, some antibodies that can't get outside, that will be stuck inside the brain and have a more efficient effect. I mean, the remanence of the drug inside the brain, that could be even more efficient.
Is there another therapeutic area in which this particular technology may prove itself before we get to the Alzheimer's, which is perhaps the largest therapeutic area of challenge? Is there an opportunity to sort of test this out in a smaller therapeutic area? I'd be curious. Dr. Wang? Dr. Chang, you want to? I don't know. I just, what I see is some capital issues here, right? You're going to need to draw capital, to John's point, you need to draw capital to this field. Is there another sort of therapeutic area?
Well, there is a huge field for brain tumors, actually, and we're showing some improved progression-free survival and improved overall survival on patients when using ultrasound concomitantly to intravenous chemotherapy. So we have somehow proof of concept that it is useful.
I think at this moment, the aducanumab is not a solution because, as we know, the Alzheimer's disease many different pathology. At this moment, the best solution is, we need to know the benefit of the focus sound first, and then we can combine many different drugs. That is my expectation in this field. But in the animal studies, combining the amyloid or tau antibody showed all the benefit. So no one knows in the real futures. Yeah.
So let me just ask each of the panelists just to sum up in the next 30 seconds with what is the greatest challenge you see in the next 2 to 3 years in this field?
I think ultrasound is going to... Oh, sorry, go ahead.
Quick comment? And that is, it is in Canada, and I'm sure elsewhere, as you know, it is approved for essential tremor. And that's an ablation approach, right, as opposed to opening up the blood-brain barrier. So, and that's like a huge difference to people who have severe tremor. It's usually still only unilaterally delivered, but that's transformative. It's changed the lives of people. So we have to acknowledge that. But here we're talking about maybe making more focused delivery of therapies for brain tumor and for neurodegenerative diseases. There could be other neurodegenerative diseases that can be focused on. There's TDP-43, which is associated with a frontotemporal dementia, often coexists with Alzheimer's disease in about 40% of postmortem cases. And that's being targeted by an oligonucleotide which again has to be delivered in a way that's a bit complicated. So I would say that we're still at the beginning phases of having antibodies that might work, that could be combined with the microbubbles to get into the brain more focally. The question is, do you go after the areas that are most damaged, or do you go after areas that are less damaged? Because we know that the default mode network and the areas where the cortical thinning, the signature areas, are already the most damaged. But can we rescue those areas because they're so important for brain function? The default mode being the state of play when you're ready to do something. And we know that and that's why I was very interested to see that we were modulating it a little bit, just by implementing the rug barrier without giving it. So I think there are, we have to kind of continue to, pursue this very carefully, with safety always being uppermost. And the development of the helmet, which, as you know, Clever Hennigan is working on very industriously, so that there's a precision helmet that a person would wear, and you don't have to be in the scanner at all. Once you've got your brain coordinates, and that would be a development that could be delivered with injection or into transfusion somewhere. So I'm not giving up on it yet, but I think it's complicated, and I think we have to understand the biology as best we can and not do harm.
With that, I think our time is up, so I am going to apologize to the panelists for not getting in the last question, but thank them very much for what they do every day. Quite frankly, that's what we patients love to see, is the commitment and the passion and the innovation that's happening in this field, and look forward to coming back year after year and seeing the progression of focused ultrasound at this therapeutic area. So thanks to the panelists for their time today. Thanks, John, for co-moderating. And thank you very much, Suzanne, for asking us to be here.
Thank you to all the panelists. We really appreciate your time and your expertise, and a very special thank you to George and John for taking the time to be here. And I'm going to take you up on that. You're coming back to every meeting after this one. Okay. So, so that's the end of the morning session. We're going to take a 90-minute break, so be back here at 1:30, for which we'll have the keynote address by Jeff Martha, the CEO of Medtronic. Remember, on the virtual platform, to look to the bottom of the page to click for the next session, or click on it on the tiles. Remember, all abstract presentations are available to view on the platform and interact with the presenters on the discussion tab. And importantly, there's a lunch session sponsored by AIUM. It's an accreditation lunch. It's downstairs in the Linden Oak Room, which is at the very end of the hallway downstairs. Food will be available outside the room, and this is important because AIUM, we want to learn about their accreditation process in diagnostic ultrasound and how that can translate into potential challenges and opportunities in developing accreditation standards for focused ultrasound. And if there is sufficient interest, the FUS Foundation is willing to support a joint working group to explore the collaboration with AIUM in developing standards for our community, which will be obviously very important moving forwards. So have a great lunch, and we'll see you at 1:30.